everyone, and welcome to today's live broadcast, Can CADD drive GPCR drug design? Rapid estimation of relative binding affinities based on pre-computed ensembles, presented by Alexander MacKerell, Ph.D., Grollman-Glick Professor of Pharmaceutical Sciences, Director, Computer-Aided Drug Design Center, University, Maryland School of Pharmacy. I am Christine Mahalik of LabRoots, and I'll be your moderator for today's event. We are delighted to bring you this educational web seminar presented by LabRoots. LabRoots is the leading scientific social networking website and producer of educational virtual events and webinars. Before we begin, I'd like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want at any time you want during the presentation. Just click on the Ask a Question box located on the far left of your screen and type your questions into the drop-down box that appears on the screen. We'll answer as many questions as we have time for at the end of the presentation. If you have trouble seeing or hearing the presentation, please click on the Help Desk button located in the promotional board at the bottom center of your screen, or use Ask a Question box and let us know that you're having a problem. This presentation is educational and thus offers continuing education credits. Please click on the accreditation button located in the promotional board at the bottom center of your screen and follow the process to obtain your credits. Please join me in welcoming Dr. MacKerell. I will now turn the presentation over to him.
Hi. Hello, everyone. Thank you for joining us today, and I will give you an overview of the methodologies that we've developed in the lab to facilitate computer-aided drug design and how we've extended those methodologies to be useful for G protein-coupled receptors, GPCRs. Before I can start my talk, I have to give you my speaker disclosure form as required by the university. One of the areas that I'm interested in is entrepreneurial activity. We have a company, SilcsBio, LLC, which is marketing the technologies that I'll be presenting today. We then do other work with the University of Maryland, different societies in the area of drug design and so on. Now, computer-aided drug design. Computer-aided drug design can be used in both a qualitative mode and a quantitative mode. Qualitatively, the scientist just wants to get an idea of what types of modifications of a compound can lead to improved biological activity, improved affinity, improved specificity, improved bioavailability. And for a synthetic or medicinal chemist, at the same time they're thinking about how to change the structure of the drug, they could be thinking about how to actually synthesize that drug. CADD methods can also be used in a quantitative format where you actually want to make predictions of ligand affinity. Will the modifications lead to improved binding, improved affinity, improved bioavailability? And we want to be able to make those quantitative evaluations in a fashion that will lead the drug design process. And if one thinks in terms of a medicinal chemistry program in an industrial setting, typically 100 or more compounds can be synthesized and tested per week. Accordingly, the CADD methodologies need to evaluate about 10 times as many compounds. So therefore, our CADD technologies need to be able to go through thousands of compounds on a weekly basis. And the methodologies we've developed allow us to do that. And the methodologies are both useful qualitatively, especially the Silks methodology that I'll introduce, and then quantitatively to allow us to make predictions of the changes in affinity for literally thousands of compounds on a weekly basis. Now, the foundation for the CADD technologies that we use are what's called molecular dynamic simulations. Molecular dynamic simulations are tools that basically allow for molecules to move in a computer. We can take the positions of molecules, we can calculate the forces acting on those molecules, and then they start to move. And once we start to move, we can use the motion combined with other equations to estimate the free energies of binding, the changes in the free energies of binding, and then also those motions allow us to generate ensembles of conformations, which then facilitate the actual calculation of the binding free energies. So we'll talk about molecular dynamic simulations, but in simple terms, we're basically allowing for molecules to move in the computer much the same way that they move in the real world. Now, for quantitative CADD, we want to be able to determine the absolute binding affinity of a protein-ligand complex or the relative affinity. Now, to calculate the absolute binding affinity, we actually have to calculate the equilibrium between the protein-ligand complex and the protein alone in solution. And we can do this with molecular dynamic simulations, but it's very expensive. These are very long simulations, which would take days, weeks, or months. And we need to do that for every new ligand. Every time we change the structure of the ligand, we have to go back and redo this huge calculation. Now, one of the ways that we can get around this is determining the change in the binding free energy, or delta delta G. And we can do that in what we call free energy perturbation, where what we're doing is we're actually perturbing the structure of the molecule from lead compound 1, say adding a methyl group or an acidic group to make compound 2, and then we calculate the free energy difference associated with that chemical perturbation. So instead of calculating the absolute binding affinity, we calculate the free energy difference, and this calculation is much faster. However, it's still relatively slow, allowing the calculation of about maybe 10 perturbations per day on a typical computer cluster that a company has. Now, key to the calculation of this free energy difference, delta delta G, is the need for an ensemble of conformations, which are indicated by these angular brackets. So basically, with both the absolute free energy of binding calculation as well as with the change in the free energy of binding, delta delta G, the key component is getting this ensemble of conformations. And that is very expensive computationally. So we want to be able to get around that so we can do our drug design in a quantitative fashion much more rapidly. So what we're going to do in our methodologies is we're going to pre-compute these conformational ensembles. Once we pre-compute these conformational ensembles, we can calculate the change in the binding affinity, delta delta G, very rapidly for many, many chemical modifications of the compounds. So we're going to do one expensive calculation up front, which will take on the order of 12 hours up until a week or two, depending on the method and the size of the protein. But then once we've got that pre-computed ensemble, we can use it very rapidly to estimate the change in the binding affinity of hundreds or thousands of modifications of our compound. And the two methodologies that I'm going to talk about today are, one is single-step free energy perturbation, or SSFEP, and the second is site identification by ligand competitive saturation, or SilcsBio. The majority of the talk will talk about the SilcsBio methodology, as this is useful for both qualitative and quantitative drug design, though we will initially introduce the SSFEP methodology, as well as later in the talk show an example of its use in the context of GPCRs. Finally, I'd like to introduce what we call the CHARM general force field and its associated program, which is important because in the computer we have to create all of the molecules. And when you're doing computer-aided drug design, you have to rapidly create the information associated with the hundreds or thousands of different chemical structures you want to work with. And the CGenFF program allows us to do that. And it's a very important component of our CADD methodologies, which works in conjunction with the SSFEP or the Silcs technologies. Okay, so now single-step free energy perturbation. Now in single-step free energy perturbation, we go in and we do our pre-computation, which is performing an MD simulation on our lead compound protein complex. We also do this pre-computation on the lead compound in solution alone. Once we've done this computation, we can then apply the pre-computed ensemble to calculate the change in the binding of a ligand hundreds of thousands of times associated with different modifications of that ligand. And we do this in the context of what we call the exponential formalism, which is used in the free energy perturbation methodology. So our pre-compute MD simulation, depending on the CPU resources and the type of protein you're using, typically takes in the order of 12 hours. Then once we have that pre-computed MD ensemble, we can then calculate our the change in the free energy of binding in about 10 CPU minutes per modification. And if one thinks about you having a computer cluster which has, say, 50 or 100 individual CPUs, you can see how quickly you can get in a matter of hours thousands of modifications. And this is the power of the approach, is pre-computing the conformational ensemble and then doing large numbers of perturbations or chemical modifications and then understanding the free energy difference. So again, just to sort of reiterate in this slide is we're going to take a protein ligand complex, we're going to do an MD simulation, and we're going to save the structures from that MD simulation. We call these the coordinates. We're going to do the same for our ligand in solution. And then once we have this collection of coordinates, this conformational ensemble, we're then going to go in and build modifications onto the parent ligand and then rapidly understand the change in the free energy of binding of the ligand by doing this calculation, which we show in the equations at the bottom of the slide, where we calculate the energy of the unmodified ligand, L0, and the energy of the modified ligand, L1, many, many times for all the different coordinate sets, all the different conformations in the ensemble. We then apply the exponential formula, which we see there, RT log, and then we see our angular brackets, to get the free energy difference in the presence of the enzyme or protein, as well as in water alone, and then we take the difference in the free energy in the presence of the protein, subtract that for the ligand in water alone, and then we get the change in the binding affinity. And again, we can do this quite quickly, and we can do it for large numbers of modifications quite rapidly. In the next couple of slides, I'm going to just show you some validation, and then later in the talk, I'll show you an example where we use SSFEP in the context of GPCRs. And when we initially developed the methodology, we wanted to make sure that we could calculate the relative binding of hydration-free energies. This is the energy of putting a molecule in solution of benzene analogs. And this would be analogous to modifying a drug-like molecule and understanding how its solvation energy changes. This is information that can be used for, say, bioavailability and so on. And what you simply see in the two plots is a nice correlation between the delta-delta-G values from our SSFEP method on the y-axis, and then the delta-delta-G values measured experimentally on the x-axis, or via a much more expensive free energy perturbation methodology called thermodynamic integration on the right panel below. And this is just validating the methodology. In the next slide, we also validate the methodology in the context of the relative binding affinity of a series of inhibitors of the protein thrombin. Again, you can see that the correlation between the delta-delta-Gs for the SSFEP method and the experimental values are quite good, giving us a predictive index of about 0.6, 0.7, which is indicating that the quantitative information predicting which modifications will increase the binding affinity versus decrease the binding affinity is quite good, and therefore this is the type of information we want to get out of this type of methodology. Okay, we'll now jump over to the SilcsBio methodology, the site identification by ligand competitive saturation. And in this methodology, what we're doing is we're calculating the functional group affinity pattern of the entire three-dimensional space in and around a protein. And this is done using a combination of MD simulations with a methodology we call grand canonical Monte Carlo, and I'll introduce that a little bit later. And in this methodology, we include protein flexibility, we include explicit solvent. This means the actual water molecules are present. And then we actually include explicit solutes, which are representative of the different types of functional groups. So what we do is we do these long GC-MC-MD simulations from which we obtain our pre-computed ensemble of conformations, which allows us to calculate what we call frag maps, which I'll introduce shortly, which we can then use for both quantitative and qualitative ligand design. And we'll go through the details of how those are used and so on. Now, in the Silks methodology, what we're doing is, again, we're doing this all on a computer. And if you show to the left, if you take a box of water, which we can have in a computer, and we say we add a benzene molecule, and we do our molecular dynamics simulation, that benzene molecule will diffuse all throughout the simulation box, and we can then calculate the probability of the benzene being in any pixel in our simulation system. And because it's only water and benzene, the benzene distribution will be the same anywhere, and if we sort of color code our distribution, the color is obviously the same throughout the entire box. It's a homogeneous probability distribution. Now, we want to understand what's happening in the presence of the protein, so we see to the right where we add a little protein to our simulation system. We then do our molecular dynamics simulation. The protein's moving, the benzene's moving, the water molecules are moving. And when you now calculate the probability distribution of the benzene, if there's a pocket on the protein surface where the benzene favorably binds, you'll have a much higher probability. We see that in red. Or there's regions where the benzene doesn't like to bind or cannot overlap with the protein, we see a much lower probability, and we see those as lighter colors, white. Now, what we do is we take the probability distribution in the presence of the protein, we normalize it, divide that by the probability distribution in the absence of the protein, and we end up with this normalized probability distribution, which we see to the lower right, where we have a probability of 1 out in solution saying there's the probability of the molecule being in solution is equal to if there was no protein there at all. We have higher probability regions where the solute, in this case benzene, wants to spend a lot of time, and then we have the lower probability regions where it wants to spend much less time, much less than being actually in solution alone. These are unfavorable regions. So this probability distribution can now go from, we're looking at it in 2 dimensions. We're ultimately going to start to convert it to 3 dimensions where we can now actually look at the distribution of different functional groups in and around a protein, and we'll start to look into that in more detail. Now, in practice, we don't have just one solute molecule, benzene, for example, in our simulation system, but we have up to 8. And here you're seeing an example of 8 solute molecules representing different types of functional groups. We see benzene, which is aromatic, propane, which is aliphatic. We've got formamide, which represents hydrogen bond acceptors and donors. We have methanol, which is an alcohol. We have acetaldehyde, which is another type of an acceptor. We have imidazole, which is an example of a heterocycle hydrogen with a hydrogen bond donor and an acceptor. And we have a negatively charged acetate, and then a positively charged methyl ammonium. So this mixture of solutes represents all the different types of functional groups that we typically see: hydrogen bond donors, hydrogen bond acceptors, hydrophobic groups, and then charged groups, and so on. And we take this mixture, we put it into a solution with our protein, and then we do a collection of these GC-MC-MD simulations. And this is our pre-computed ensemble from which we're going to get what we ultimately call our fragment maps, and I'll introduce those in a moment. In addition to the solutes that I'm showing in this slide, we can also have a collection of solutes which, say, includes different halogens, chlorobenzene, bromobenzene, fluorobenzene, trifluoroethane, and so on. So we can have halogen frag maps. And halogens are known to exist in a large percentage of drugs, and they accordingly are of interest to drug design, so we can have these halogen fragment maps and use those and so on. Okay, so basically what I'm showing here is the actual MD simulation of a protein. It's in a box of water. You see the water molecules are shown in red, And then we have our different solutes, which are floating around in there. You can see the different solutes floating around surrounded by the water molecules. And basically, this is our pre-computation, where we're doing these MD simulations. And what we then do is we put the system into a grid, and we calculate the probability of the different types of solutes, the different types of functional groups being in the different grid points. I'm showing this in 2 dimensions, but once we actually have a protein surrounded by water in 3 dimensions, and this ultimately allows us to calculate the 3-dimensional probability distributions for the different types of functional groups in and around the entire protein. I'm showing those probability distributions to the right, overlaid on the protein on which we were doing the MD simulation. So this is sort of the basis of the Silks technology, is this pre-computation from which we get these frag maps, these functional group probability distributions, and we can now use these frag maps both qualitatively to visualize where we want different types of functional groups, as well as quantitatively to estimate the change in the binding affinity of different ligands. And I'll follow up on this in the next few slides. Looking a little bit more closely, we're looking now at the example of frag maps on p38 MAP kinase. And what you see here are the different types of fragment maps. They encompass the entire protein. In this case, with p38 MAP kinase, we can see how the center of the protein is the binding site that we're interested in. But what you see now is you see the different frag maps in different types of regions telling us where we would like to have, say, hydrogen bond acceptors, hydrogen bond donors, aliphatic groups, aromatic groups, and so on. So this is, again, the basic information that we get from the Silks pre-computation, and then once we've got this information, we can use this over and over again for any ligand that we're looking at, and so on. Now we're going to focus in on the binding site of the p38 MAP kinase, and we've now overlaid an inhibitor of the protein from a crystal structure onto the maps. Now, if you look at this figure, what you see here is the binding site. We see the solvent accessibility of the protein associated with the crystal structure, and then we see the different fragment maps. And if you look at the fragment maps up to the right, we see these aliphatic and aromatic fragment maps, and they're hydrophobic groups, so they correspond with each other. Interestingly, in the center there, we also see a hydrogen bond acceptor map. So this is indicating that in this region, we could add either aliphatic groups, aromatic groups, or hydrogen bond acceptors to a molecule to potentially improve the binding. Also, what you see here is the maps actually penetrate underneath the surface of the protein. And this is important because in these MD simulations, we're allowing the protein to move so we can actually understand how the protein structure can open. It can change its conformation to allow for ligands to bind. And this is important because when we're doing our drug design process, we're no longer just limited by the surface of the protein from the crystal structure, but we can start to think in terms of the way that the protein structure can actually change to accommodate different ligand modifications and so on. And we'll look at that in a little bit more detail too. This is exemplified in this slide, where to the left panel, again, we show this similar view as we saw before, where we see this solvent accessible surface. I've sort of shown more of the solvent accessible surface. The region underneath the surface, you can see here to the right and to the left in the left panel. And now I'm showing in the right panel what we call the exclusion maps. And these are the regions of the protein where the solutes or the water molecules during our MD simulations can never access. They're excluded from accessing those regions. And what this is doing is this is giving us an idea of the full range that the protein can open. So if we look to the maps, the solvent accessible surface to the left, and then we compare that to the right, we can see how much further back the exclusion map is. And so this entire region, which would be essentially underneath the solvent accessible surface, this can actually be accessed when we're doing ligand design. So we can actually think it in terms beyond going beyond the protein surface, and we can judge this based on the exclusion maps. So in the end, when we're actually doing our ligand design, we basically get rid of the protein structure, and we just work in the context of our fragment maps and these exclusion maps to allow us to think about how to modify the ligand, to create modifications that will be synthetically accessible, and then to quantitatively evaluate those modifications to understand which of those will lead to improved binding and so on. What's also interesting about the Silks methodology is we include the water molecules, and that's important because when you're doing ligand design, there's certain water molecules that will bind to the protein and be very difficult to displace. And if you're trying to modify a ligand to occupy a region where you can't remove a water molecule, that ligand won't bind well. So what I'm showing here is, to the left, I'm showing in red spheres the position of water molecules from a crystal structure. And then the red 3D maps are the positions of water molecules from our Silks MD simulations. And you can see in many cases the crystal water molecules and these maps correspond quite well. But what we really want to know is which of these water molecules can our functional groups compete with? Can they remove, allowing us to add those functional groups to the protein? And we get that information from the slide on the right, where I'm showing the same view. I still have the crystallographic water molecules. I have the fragment maps associated with the water molecules, but then you can see which of these water molecules also overlap with different fragment maps, say with an aliphatic map, with say a positively charged map. And this is telling us which of these water molecules functional groups can compete with, so we can add that functional group. We're confident that it will be able to displace the water molecule and therefore lead to improved binding affinity. In other cases, you can see the water fragment maps with no other map there. That indicates that these water molecules will be difficult to displace. And again, we don't want to try to build our molecule out into that region, because the water molecule, it would be unfavorable to displace that, and therefore our ligand would not bind well. Now, those are some ideas of how we can use the maps qualitatively. We can also use them quantitatively by converting the Silks frag maps from probability distributions into what we refer to as grid-free energies. This is basically a Boltzmann transformation of the normalized probability distributions, which we can also refer to as occupancies, from which we now obtain free energies. And once we've got these free energies, we can assign free energies to the individual atoms in each molecule, sum up the grid free energy contributions for each atom in the molecule to get what we call the ligand grid free energy. And this is actually a proxy for the binding affinity of that ligand. It's not exactly the same as the experimental binding affinity, but we can use this to rapidly estimate the relative binding affinity of different ligands, different ligand modifications, and so on. Now, once we're working in the context of the grid-free energies, we can now start to look at our fragment maps at different contour levels. And what I'm showing here are now aromatic frag maps at contour levels of -0.5, -1.0, and -1.5. And you can see as we go to more favorable energies, we focus in on the regions where, in this case, an aromatic group will bind most favorably. And then as we go to less favorable and even less favorable, the maps spread out more, and they're showing us the full range of possible regions where aromatic groups can bind. So if we added an aromatic group in the upper right panel to sort of that region to the left, we would still have an improved binding affinity, although it would be much less of an improvement than in the region to the lower right that we see at the bottom of the slide. So now we can think in terms of energies, and this is very important for the ligand design, because we can now start to understand the energetic contributions of the individual atoms in each molecule. And here I'm showing a inhibitor. Again, this is an inhibitor of our p38 MAP kinase, which is overlaid on the fragment maps. And then I can understand the energetic contributions of the different atoms in the ligand to the overall binding affinity. Sorry, the font's a little bit small here, but you see the central imidazole functional group. the free energy contributions are relatively small, whereas the free energy contributions are much larger in the vicinity of, say, -2, indicating that the overall contribution of, say, this aromatic group is approaching a couple kilocalories to the overall binding affinity, and so on. Also, we can think in quantitative terms where we see that the different types of functional groups are overlapping with the corresponding fragment maps. We see the positive fragment maps up here overlaying with a positive functional group, the aromatic and aliphatic maps overlaying with these aromatic ring. We see acceptor maps, which are overlaying with a fluorine, which acts as a hydrogen bond acceptor. Down here, we see acceptor maps overlaying with the acceptors in this ring system. Also, we see that this amino group is actually not overlaying with any maps, indicating that its contribution to binding is relatively small. And then importantly, when we look at these maps, we see the maps beyond the molecule itself, and this is giving us information on how to extend the molecule to increase its binding affinity. And so if we look at this ring system up here, we can see adjacent to that are aliphatic and aromatic maps, as well as donor maps. This is indicating that maybe we can add a aliphatic group, which includes maybe some type of a hydrogen bond donor, maybe an alcohol, and that would improve the binding affinity. So we can look at these maps in a qualitative way, as I've been referring to get some ideas of the different types of modifications. And then because we're working in the context of these grid-free energies, we can now start to make quantitative estimations of how the binding affinity changes and so on. And once we now have these grid-free energies, as I referred to before, we can sum over all the grid-free energies for all the atoms in a molecule to get the ligand grid-free energy. And here I'm showing some thrombin inhibitors where we have some ligand grid-free energies, and we can see how they correlate with the actual known binding affinities of these molecules and so on. Also, once we've got these grid-free energies, we can take our molecule and we can use what we call Monte Carlo sampling to allow the orientation of the molecule to relax in the field of the fragment maps, allowing it to adjust its orientation and so on. And so this is a very powerful technology to allow us to understand how different types of modifications will change the local conformation of the ligand. And this is important for understanding how it binds and so on. Now, let's move to GPCRs. Now, GPCRs, G protein coupled receptors, there's, I think, an order of estimated about 3,000 different types of GPCRs in the body. A large number of drug molecules target GPCRs. There's a huge amount of effort to understand GPCR structure function, correspondingly to develop therapeutic agents against GPCRs. Now, what I'm showing here is actually the outside edge of a GPCR. And one of the interesting things with GPCRs is different types of molecules bind to different regions of the GPCRs. And this is a challenge for computer-aided drug design in general, is identifying and characterizing alternative binding sites to GPCRs. The typical binding site of a GPCR where the ligand for that GPCR, say dopamine to the dopamine receptor binds, is called the orthosteric site. We want to be able to quantify non-orthosteric binding sites, and I'm showing several of those sites where lipids or cholesterol bind to the surface of the GPCR. To the left and to the right, this is where polyethylene glycol, PEG, binds on the pocket to the surface of the GPCR. So this is one of the challenges of computer-aided drug design for GPCRs. Now, when we look at a GPCR, what I'm showing to the left is the mu-opioid GPCR, and these G protein coupled receptors are made up of a series of transmembrane helices. So here's the structure of the protein. It would be in a lipid bilayer, would be surrounding that protein, and then the ligand for this protein, in this case a analgesic agent referred to as BU72, is binding down deeply into the GPCR. Now, if I take this GPCR, which I'm looking from the side here, and I look from the top, and I look at what we call a solvent accessible surface, we can see that a little bit of the binding site, but that binding site is very deep, and it's partially inaccessible. And this is a challenge to the Silks methodology because we have to make sure that our functional group maps, our frag maps, can penetrate into that deep pocket to give us the information we need to do protein ligand design in the context of GPCRs. So the limitation of the Silks methodology when we initially developed it was only using molecular dynamic simulation, was having our solutes sample into these deep inaccessible pockets. And when you're doing an MD simulation, your molecules are diffusing around in solution into the different binding pockets, out of the pockets. And this rate of diffusion is limited, especially if you have to move into a very deep pocket in the protein and then move out again. And also, we have to worry about big conformational changes that need to occur in a protein because our MD simulations are limited in time, such that we've got to make sure that we don't we're not limited by large conformational changes in the protein that can make the binding pockets, say, inaccessible. And while I'm focusing on GPCRs today, this type of problem is also relevant for other types of proteins. For example, these nuclear receptors, and I'm showing a picture of the androgen receptor here. And the binding pockets of the androgen receptor and other nuclear receptors are totally blocked from the protein surface, since the protein has to partially unfold. And again, we wanted our Silks maps, our frag maps, to be able to sample these very deep or totally blocked binding pockets so we can use the technology for GPCRs and other types of proteins with these very deep inaccessible pockets. So what we developed to do this was the grand canonical methodology. And the grand canonical methodology is a well-known simulation methodology. It's been known for a century or more. But basically, in this methodology, the trick here is instead of just taking your simulation box and fixing the number of atoms in there, you actually allow the number of atoms to change. And we call this constant chemical potential. So we can have, say, water molecules come into the box, move out of the box, and so on. And we can do this in a way that corresponds to a correct energetic ensemble, a correct thermodynamic ensemble, so we can get free energy information in the context of moving water molecules or solutes into and out of the box. And to think of the GCMC methodology, we call now Grand Canonical Monte Carlo methodology, we think of our simulation system. In this case, we've got a sphere. We usually have a rectangular type of system where we have like a bath of, say, water molecules, and we can take those water molecules and move them into the simulation system or move them out. And what we do is in the Grand Canonical Monte Carlo methodology, we assign a chemical potential, in this case 5.9 for water, and if you started to move the water molecules in and out at equilibrium, the concentration of water would correspond to 55 molar. Analogously, if we had a box of water and we took a solute, say benzene, and we assigned its chemical potential to correspond to that of its free energy of solvation, and we did our Grand Canonical Monte Carlo simulation, the benzene, our solute, would end up at a concentration of about 1 molar in our simulation system, where this corresponds to what's called the standard state. However, it's been known that using this Grand Canonical Monte Carlo methodology is very difficult for molecules larger than water. And what we developed to deal with this is what we call an oscillating chemical potential, where chemical potential is indicated by μEx, Grand Canonical Monte Carlo methodology. And basically, if we have trouble moving a solute into the simulation system, we basically just turn up the chemical potential. We give it more energy to do that, in essence. This is rather technical. I won't go into more of the details here. I'll leave that for the reader. There's the relevant publication. But this is now allowing us to use this GCMC methodology to start to sample these deep, partially inaccessible pockets in proteins such as GPCRs to extend our Silks methodology to those types of systems. So what we end up doing now is we end up using the GCMC methodology to sample the distribution of solutes in water throughout the entire simulation system. And then we follow that with our molecular dynamic simulations that allows for conformational sampling of the solutes in and around the protein, as well as for the conformational sampling of the protein itself. And this is important because remember, as we talked about earlier, we want to see how the protein can open to allow for understanding how the protein can change its structure to use that in drug design and so on. And in more, again, this is more technical terms, we use the GC methodology for, say, 200,000 steps. We follow that with a nanosecond of molecular dynamic simulation, and we repeat that typically for like 100 iterations, and we actually do that for 10 different simulations. So in the end, we use our GCMC-MD methodology to perform about 1 microsecond of molecular dynamic simulation. And again, this is all our pre-computation. And this pre-computation typically takes in the order of a week or two, depending on the MD, the size of the MD simulation system, the size of the protein. But then once this is done, we have our fragment maps, and we can now use them over and over again in ligand design. And I'll go into some more examples of that as we go through the talk. Just a slide on validating the methodology, where we applied the method to a protein called T4 lysozyme, which has a pocket which is totally blocked from the surface of the protein. And what we can see here is that we have a nice correlation between our ligand grid free energies from the Silks methodology and the experimental binding affinities. And we see that with 2 different approximations with respect to how we calculate the ligand grid free energies. So again, this is just supporting the quality of the methodology for quantitatively predicting the relative affinities of different ligands bound to a protein. So we get back to now our GPCRs, and here I now show the Silk simulation setup for the mu opioid receptor. You can see here's our GPCR. It's got the 7 transmembrane helices. Here's the extracellular face. Here's the intracellular face. Our ligand binds down in here. In the simulation system, we also have our lipid bilayer, which in this case is made up of POPC, along with cholesterol molecules. We have about 10% cholesterol. Also, you see the water molecules above and below the protein and lipid bilayer. And then in the water shown in red, we have our different solutes, which are used to calculate the functional group distributions or the fragment maps. So now we go back to our problems. Now, we can now show that we can apply the Silks GCMC-MD methodology to the opioid receptor. We can now look into the binding pocket, and you can see the ligand. But now we can see that the ligand is encompassed in different types of fragment maps, showing that we've effectively sampled different solutes down in this deep pocket. So we now have the frag maps, and we show to the right the frag maps actually overlaid on the ligand from the crystal structure. And you can start to see how the different types of frag maps correspond to the different functional groups. You see both acceptor and nonpolar fragment maps corresponding to this ring system, to this ring system. The acceptor maps correspond to this oxygen, as well as to this hydroxyl. Up here we see positively charged maps corresponding to the positively charged nitrogen, which is typical in opioids, and so on. So now this is the type of information which we can start to use for both our qualitative and our quantitative ligand design, where we can understand how we can sort of extend the molecule in these different directions to improve its binding affinity and these types of things. So we can now, so this is again just showing how we've extended the Silks methodology to allow it to be applicable to ligand design against GPCRs. In this case, we're using the opioid receptor as an example. With respect to identifying alternate binding sites, here are these sites on the surface of the GPCR. Here is the crystal structure of this polyethylene glycol bound to the protein surface. And here we've got the fragment maps overlaid on the surface along with the ligand. And what you can see is how nicely the fragment maps have identified this putative binding pocket. And you can see they also identify other types of binding pockets on the protein surface. So the Silks methodology just doesn't focus in this case, the orthosteric site associated with the agonist binding, but is basically mapping out all possible binding sites in and around the entire protein. And this is very powerful. In the next slide, we show the image of the two lipids and the cholesterol bound to the protein from the crystal structure. We again see how nicely the fragment maps map out the location of the lipids, especially their aliphatic hydrophobic tails. And what was interesting here is the fragment maps did not identify the cholesterol binding site. And what was happening here was we actually had cholesterol in the simulation system, and the cholesterol went in and bound to this site, and it bound there so tightly that the other solutes can never compete with this. And this is suggesting that this cholesterol molecule potentially has a functional role in the biological activity of the opioid receptors. And this is just a hypothesis based on the results from these simulations. But again, because we have the full simulation system with the lipid bilayer, the cholesterol, we can learn these types of, we can make these observations to allow us to sort of not just do drug design, but also to think about how GPCRs function and so on. Now, I'm going to follow these results based on the opioid receptor with a study that we've done on the Beta-adrenergic receptor, which is again another GPCR. This is an interesting system to apply methodology because we have crystal structures both in the presence of agonists and in the presence of antagonists. So we have an active form structure and an inactive form. And with GPCRs, we know that there's conformational changes that occur such that the relative orientation of these transmembrane helices shift, and these control the active versus the inactive state and so on. There's binding affinity for a large number of ligands. In the context of therapeutics, the beta-adrenergic receptor is the target for agents for the treatment of asthma, such as isoproterenol, where if you agonize the B2-adrenergic receptor, it'll lead to bronchodilation. However, these compounds, which are out on the market, have problems associated with tolerance and desensitization, which motivates identifying novel adrenergic agonists that will have lower tolerance. So this motivated this therapeutic study. So we went in, we're going to apply the Silks methodology. Again, here's our simulation system where we have our GPCR in the center in green. We have the lipid bilayer with cholesterol. We have the water molecules and the solutes in the top and the bottom. And for this particular system, we focused on the binding region for the GCMC. However, we did MD on the entire simulation system. We went in, we calculated our pre-computed ensemble from which we've got the fragment maps, and then we started to use the fragment maps for our ligand design. In the next slide, I'm showing the fragment maps where we can see the fragment maps for the active conformation of the protein. We can show the fragment maps for the inactive conformation. And what we see here is that there's significantly differences in the fragment maps for the active conformation versus the inactive conformation. And this is important because we can now take the difference between these maps to identify regions where, if we say occupy those regions, we'll have an agonist versus an antagonist or vice versa. So we can see how by doing two sets of Silk simulations, calculating two sets of fragment maps in the presence of, again, these different conformations, we can get difference maps, which we can then use for ligand design to specifically design agonists versus antagonists. So we can think of terms of activation as well as improved binding affinity and so on. And this is quite useful in the context of GPCRs to get agonists versus antagonists, but we've also used it for different types of proteins such as MCL1, the BCL2 family proteins, MCL1, BCLXL, to identify ligands, say, specific for one protein versus the other protein, or ligands that will bind to both proteins and so on. This is an example of this has been published a couple of years ago now. Now, you can see, again, we're visualizing the maps, so qualitatively you can see how this facilitates your ligand design. Quantitatively, we can validate the usefulness of the active maps versus the inactive maps, where what we see is if we take the active maps with a series of agonists or partial agonists, we get nice correlation. However, if we take the inactive maps with the same series of agonists or partial agonists, we get much poorer correlations. However, if we take inactive maps with antagonists or inverse agonists, we get good correlation. However, those same, antagonists or inverse agonists with the active maps give poor correlation. So this idea of looking at different types of frag maps for the active state and the inactive state can be used quantitatively, as we're showing here, as well as qualitatively, as we showed in the previous slide, and so on. So what we wanted to do here is come up with totally new agonists of the beta-adrenergic receptor. So what we did is we applied what we called the Silks pharmacophore methodology, where what we can do is we can take the fragment maps and we can convert them into pharmacophore features. And you can see this would correspond to, say, a positively charged maps, hydrophobic maps, hydrogen bond donor maps, and so on. And what we can do is we can then do a database screen, a computational database screen, to identify compounds that will have the different structural features which we've determined from the Silks fragment maps and then converted it into what we call a pharmacophore. Now, this allows us to go in and screen this large database. In this case, we screened a database of about almost 2 million compounds. And from that, we identified the top 50,000 compounds whose structural features match that of the pharmacophore, at the different types of hydrophobic groups, hydrogen bond donors, negatively charged, and so on. We then took those top 50,000 compounds, and then we screened them in a couple steps, including using the ligand grid free energy values from the Silks MC. And what we did here is we looked at the energetic difference with respect to the active form of the protein versus the inactive form. So we could then focus in on going from, say, 50,000 molecules down to 1,000 molecules, down to 100 molecules, where we're now identifying those molecules which will have a high probability of binding to the receptor, but then will bind to the active form versus the inactive form. And then from this 100 compounds, we selected a collection of 15 compounds based on chemical features. And what we want to do in a drug design study at this point is maximize the chemical diversity, because we want new leads which are structurally different than known compounds and so on. We then took those 15 compounds. Our collaborators at Thomas Jefferson tested those compounds, and 7 of our compounds were actually agonists, which is a very good hit rate for these types of virtual screening methodologies. And we can see we're comparing those with the compound isoproterenol, which is a therapeutic agent out on the market. So we've moved the study, the methodology to this point. We have these compounds, and this is, we've happily just recently got this study funded, so we'll be able to move this ahead in the near future. Now, what I also wanted to do with the beta-adrenergic receptor is show how we can use the SSFEP methodology. We're going to go back to SSFEP in the context of a GPCR. So what we're going to do here is adrenergic receptors bind different types of small molecules, these biogenic amines such as dopamine. So we set up a model calculation where we're going to take dopamine, we're going to bind it to the adrenergic receptor, and then we're going to apply the SSFEP methodology, and then we're going to look at a large number of modifications to identify those modifications that are predicted to have improved activity. And this is getting back to the power of the SSFEP as well as the Silks methodology to calculate free energy differences of large numbers of modifications. And what we're seeing up here on the dopamine molecule to the right are all the different types we can modify, all the different sites in the molecule we can modify. At each of those sites, we're going to evaluate 43 different types of modifications. Of the 430 types of modifications, we actually evaluated 410 modifications, as 20 of those modifications were not structurally suitable for the ligand alone. They were just, the modifications were too big. To give you an idea of the types of chemical modifications, I'm showing them here. You can see we typically, for the SSFEP methodology, we typically add relatively small functional groups, halogens, methyl groups, ethyl groups, different types of polar groups, and so on. But again, the power of the methodology is that we can go through, we can do our pre-computed MD simulations of the protein ligand complex, as well as the ligand in solution. We can obtain our pre-computed ensemble. We can then go back and, for the 400 plus modifications, rapidly calculate the change in the free energy associated with those modifications in the presence of the protein, as well as in the presence of the ligand. We then get the change in the binding affinity. Again, this is based on the changes in the free energy in the presence of the protein and the absence of the protein, or just in water alone. And we now have hundreds of free energy differences, which typically can be obtained on an overnight basis, as we can calculate we can do the pre-computed ensemble calculation, typically on the order of about 12 hours. Calculating all the modifications takes another hour or two. And this is the power of the methodology, where we can take our GPCR, as I'm showing here, we can put it into the lipid bilayer surrounded by water molecules. We can do our pre-computed MD simulations, and then we can rapidly predict the change in the binding affinity of large numbers of modifications. And so what I see here, what I'm showing here in this slide, is again our dopamine molecule with all the sites that we've modified, 10 sites in total. We're showing results for a subset of those sites, including both R and S modifications. You see this 5A to 5B. And what we see here is that at many of the sites, all of the modifications are unfavorable. So those sites would be indicated, we don't want to do modifications to those sites because they're bad sites. However, at some sites, we do see favorable modifications, and those are the sites that we would like to follow up on when we're actually synthesizing molecules and doing testing. So you can see how we can use the methodology to eliminate certain sites from chemical modifications, as well as identify those sites which modifications are favored. And then we can actually go in and we can say, okay, we've got 5 favorable modifications. What are those modifications? What actual chemical groups are they? And actually then go and synthesize and test those. And at the bottom here, I'm showing a table where we're showing that the change from dopamine to norepinephrine experimentally leads to a favorable change in binding. And we get that favorable change in the case of the SSFEP methodology. The magnitude of the change is not as large as that is seen experimentally. But typically, when we use the SSFEP methodology or the Silks methodology, we look at the direction of the change. Is it more favorable or is it less favorable? Because that's the type of information that, say, a medicinal chemist wants to use for ligand design and so on. And in this case, with the beta-adrenergic receptor, I focused on using the SSFEP methodology. We can also use the Silks methodology to calculate the free energy change for these large numbers of modifications in a very analogous way. Some additional validation we did with the SSFEP, where we now started with epinephrine. We took epinephrine, we then did modifications of epinephrine to norepinephrine or to dopamine. And what you see here is both of these modifications are predicted to be unfavorable, and the SSFEP methodology correctly predicts those modifications, indicating the validity of the approach. And again, what we're focused on is the direction of the free energy change, using that as the vehicle to tell us what modifications to make or not to make, less on the actual quantitative. Is it negative 2 kilocalories? Is it negative 1 kilocalorie? And so on. Now, I'd just like to point out a couple other studies that we've done on GPCRs. This is a Silks based study on the metabotropic glutamate receptor subtype 5, mGluR5, where we did a structure-activity relationship on a series of compounds. We developed the Silks frag maps for this system. This was actually based on a homology model before a crystal structure was available. You can see here we have the Silks frag maps, which are synthetic collaborators were able to use to guide their ligand design. We then get reasonable quantitative predictability from the CADD methodology. Again, this is just a study that there's the reference. But what was interesting here is with our same collaborators, we were then able to perform a second study on an entirely different series of compounds, but we were able to use the same set of fragment maps and then use those to qualitatively drive ligand design, and then we can see the quantitative predictability of the methodology. And what's important here is we did not have to go back and recalculate the Silks fragment maps. I just want to emphasize again that once we've got these fragment maps for a given protein, we can use them again and again for any lead compound. They do not have to be recalculated. And again, this is the general power of the pre-computed approaches that we've developed in the lab and are now available to the scientific community and so on. And then finally, I mentioned before the CGNFF force field and the associated program, which is important because when we're doing the Silks or SSFEP methodology, we have to rapidly generate hundreds or thousands of different types of ligands. We need to generate them in the computer. The CGNFF program does that for us very rapidly. The quality of the force field has been shown by a number of workers in the field to be of high quality, of high accuracy. And this allows us to then quickly move in to do an automated workflow of ligand design, where we can go in, we can take our lead compound, we can go in, we can specify large numbers of modifications. The user typically can visualize, say, the fragment maps in the context of Silks, identify a number of possible modifications. We then generate all of those modifications very rapidly using the CGNFF technology. And then we go in and we do the Silks calculations and get the LGFE scores. And we can literally calculate thousands or tens of thousands of modifications on a daily basis once these Silks frag maps are already in place. And again, we can use the SSFEP methodology as an alternative. I'd also like to emphasize with the Silks methodology, the value of the Silks methodology is we can study much larger chemical modifications than we can with SSFEP. SSFEP is typically limited to modifications of, say, 2 or 3 non-hydrogen atoms, whereas the Silks methodology is you can look at very large chemical modifications when you're doing your ligand design and so on. So just to finish up, I'd like to acknowledge the people in my lab, Olga Vunch, Prabhu Rahman, Wenbo Yu, Kaushik Fang Yu, Lei Fang, Wenjun Keno, who worked on the various SSFEP, Silks methodologies, as well as the Charm General Force Field. Our collaborators on the Beta-adrenergic receptor up at Thomas Jefferson, Lena and Deepak, Fentian Zhu and his lab members worked on the mGluR receptor, and then various funding from the NIH, NSF, the Waxman Foundation. And then also my long support by the University of Maryland School of Pharmacy, the University of Maryland Baltimore Computer-Aided Drug Design Center, and then SilcsBio LLC, which is now the commercial arm of the SSFEP, Silks, and CGNFF technologies. All right, thank you.
Excellent. Thank you, Dr. MacKerell, for that informative presentation. We will now start the live Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click on the Ask a Question drop-down box located on the far left of your presentation window. Type your question into the box that appears on your screen and click on the Send button. We'll answer as many questions as we have time for. So let's get started with our first question of the day. Our first question asks, what are the computational CPU requirements for Silks in the SSFEP methods?
Again, it's the pre-computation that takes the majority of the time. For the SSFEP method, it's typically on the order of 12 hours. This can actually be much shorter on the latest state-of-the-art GPUs. With the Silks methodology, the pre-computation takes on the order of 3 to 4 days for a relatively small protein. It may be up to 2 weeks, 3 weeks on very large proteins. And again, we're doing 10 simulations in parallel. But again, it takes on the order of weeks for the Silks methodology. But once we've got those finished, and we have the pre-computed ensemble for both the SSFEP method and the Silks methodology, it takes in the order of minutes to evaluate each individual modification, and this allows us to do thousands of modifications very rapidly using the two methodologies once the pre-compute is finished. Thank you.
No, thank you. Excellent. Thank you for answering that question. Our next question of the day asks, what information on the system of interest is required to apply the Silks and the SSFEP methods?
All you need is a protein structure. For majority of therapeutically interesting proteins, protein structures are available from X-ray crystallography or from NMR spectroscopy. If there's not a three-dimensional structure of your protein available, there's likely a structure of a homologous protein, so homology modeling can be used to determine a three-dimensional structure, which then allows for the SSFEP or the Silks methodology to be used. In the context of specific ligand design, it's often quite helpful to have a crystal structure of your ligand bound to the protein so you know the bound orientation, which will facilitate just doing the modifications with either the Silks or the SSFEP methodology. Thank you.
Okay, and it looks like we have time for one last question today. And this question asks, what size conformational changes can occur in the protein during the MD simulations?
The MD simulations each simulation is on the order of about 100 nanoseconds. So conformational changes in the protein that occur on the order of 3, 4, 5 angstroms can occur during those simulations. If there's a very big conformational change, such as the change between the active and the inactive conformations of a GPCR, those will not occur in these simulations. Accordingly, what we've done is we'll do individual calculations on the active state and the inactive state and then combine those together for doing ligand design for agonist versus antagonist, as we showed based on the beta-adrenergic receptor. Thank you.
Well, thank you, Dr. MacKerell, for your presentation. I'd also like to thank LabRoots for making today's educational webcast possible. Before we go, I'd like to let everyone know that today's webcast will be available for on-demand viewing through May of 2018. You will receive an email from LabRoots letting you know that this webcast will be available for replay. Please share that announcement with your colleagues who may have missed today's live events. That's all for now. Thank you for joining us, and we hope to see you here again soon at LabRoots. Goodbye.